So, A'udhu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. Rabbi shrah li sadri wa yassir li amri. Wahlul 'uqdatam min lisani yafqahu qawli. Rabbi zidni ilma warzuqni fahma. And assalamu alaikum wa rahmatullahi wa barakatuh. And good evening and ayubon. Okay. So, what is the style of the paper class, right? Okay. So here below I will show you some questions based on unit number 6. Last class you may remember that we had discussed all the organelles and the function. So now I am going today in this class, today we are going to do some other model paper question. Last class we had done some past paper questions now. So today we are going to discuss some model paper question related to unit number 6, and we are going to discuss regarding the cell division today. Cell division and some question based on the cell division. Okay. So I will show you the question here, Bilal. The first question number 1. Okay. Do this question and send me the answer to the WhatsApp within 2 minutes. 2 or 3 minutes. Okay. You know the answer, right? Last class we had discussed all the names and all the functions of these questions or organelles, Bilal. Don't take more time. Don't take more time. Within 2 minutes or 3 minutes, send me the answers through the WhatsApp or the chat box. Don't take more time, Bilal. If you don't know the answer, it's okay. You can refer your textbook, Bilal, because when you're referring the textbook, what will happen? You will learn more number of things, right? When you're doing the paper, if you don't know the answer, it's okay, fine. Don't see the answer of the past paper. Just refer the textbook. Then you will learn more number of things at a time, right? Okay. Okay. It's okay, fine. It's okay. Those who have missed our last class for the unit number 6 paper class, past paper discussion last week, it's okay, fine. After this question, I will be explaining the question here. We have the explanation. I will explain one by one. Don't worry, right? Okay. Now just try to find the answer. Those who have missed our last session, don't worry. I will explain those questions later. I will explain once again. Don't worry, right? Don't take more time, Pulal. You can send me the answer to the WhatsApp or the chat box. One of you has sent me the answers. Rahamani, very good, my dears. The answer is correct, right? Okay. Shall I explain? Or do you want time? And there is the same question, Pulal. This is a model paper question, and there is a past paper question. Last week we have missed two questions. I don't know whether you have remember that one. We have missed two essay questions, structure question. The same pattern, same image. I will explain that question also, right? Little bit late. After discussing this question, we'll discuss that one, right? Then you will realize the same question has been repeated. And Jazahilmi. Jazahil. Alright, okay. Very good. Well, if it is difficult to type, if you are feeling difficulty to type, you can send me the answers through the WhatsApp, okay? And Zuha Nuha. Check the answer for the A, Bilal. Check the answer for the A. But B and E are correct. You have sent me the answers. Ashmita sent me the answers. Very good. Perfect. Yes. Mashallah. Very good. All the answers are correct. Very good. Very, very good. For the C one. Okay, very good. Answer is correct. Mashallah. Fine. All the answers are correct, my dear. Who is this? Ashmita. Very good. And Khadija from Puttanam. And Jazahilmi over the Adapring Paravan. And Khadija from Putlam. Okay. Khadija, we'll check the answers. Okay, don't worry. We'll check the answers. Sorry. Fine, plan. Now, I have given you enough time to answer the question. Some of you have sent me the answers. Some of you have didn't send the answers. Okay, fine. Now stop writing. Stop all the other works. Stop all the other works. Stop all the other works. Now listen to the screen. Okay. Look at the screen. Don't write anything. Okay, Dani. Now your time is over. Now this is my time. Okay, Dani. Don't do any other works and look at the screen. Here, question number 1. This is a structure of the animal cell. And last class we have explained the organelle, the name of the organelles. Okay. And this is animal cell. Now I'm going to show you the plant cell and animal cell side by side. This is the animated 3D structure. And this is the animated 3D structure for the plant cell. But in the exam, you will never get the structure like this or this. You will get the image like this, 2D structures in the paper. So at that time, you must be able to name the organelles by using this picture, not these pictures. These pictures are used to explain in the class, right? But in the exam papers, you will get the picture as like this 2D pictures. Okay, then. So you have the plant cell in your left hand side, and you have the animal cell in your right hand side. So my question is this, Pula. Just compare the plant cell and the animal cell. And can you find some differences? Last class I explained 4 differences, right? Okay, wait, listen to the screen. Listen to me, okay? Look at the screen here. So here we have the cell wall here, right? Cell wall. But in the animal cell, Pulal, we don't have the cell wall. Am I right or no? So here we have the cell wall and here we don't have the cell wall. And second one, the chloroplast. Look at the picture here. We have the chloroplast in the animal cell, but sorry, in the plant cell, but in the animal cell, we don't have the chloroplast. If we have the chloroplast, we will be in green in color, right? We are not green color, am I right or no? Because we don't have the chloroplast. So we don't have the chloroplast, but the plant cell will have the chloroplast. And third one, here you can see a very big vacuole. A large vacuole is there, right? Later, there is another question based on the vacuole. So I will explain the vacuole separately, a little bit later. Don't worry. Later. So in the animal cell, we don't have the vacuole. Am I right or no? If the animal cell has the vacuole, they will have only small size of vacuole, not this much of vacuole as plant cell. So we have discussed third example here. We have the vacuole, and here we don't have the vacuole. And Pulal, just compare this one. Up to now, I have said that plant will have this one, animal cell will doesn't have this one. Plant have this, animal cell doesn't have this, like that. Can anyone tell me there is an organelle that can be found in the animal, that can be present in the animal but absent in the plant? Present in the animal and absence in the plant. Centriole. Very good. Last class I explained that one, right? Here, the centriole.
Rough endoplasmic.
Pardon, my dear. Anything I couldn't hear, my dear. Okay. So in the centriole bulal, it can be found in the animal cell, but not in the plant cell. Did you get the idea? We have discussed four differences between the.
In plant cell, there's rough endoplasmic, and in animal cell, there's smooth endoplasmic.
We can't say like that, my dear. In this picture, they haven't mentioned that one, but rough endoplasmic reticulum can be found in the animal cell. Okay, in the picture, okay, you may have found that one, but we can't tell that one as a difference because in this picture they haven't mentioned that. Okay, But in the animal cell also we have the rough endoplasmic reticulum. Okay, my dear. You have compared the pictures. Okay, fine. I won't appreciate you, but this is the thing, Pulal. They haven't mentioned that, but in the animal cell we will have this one, rough endoplasmic reticulum.
Okay, sir.
Right? But they have missed that to mention that. Don't worry, yeah? But you
Okay, sir.
is it clear, my dear? You have compared the pictures and you have told the answer, but we can't consider that one as because in the animal cell we will have the rough endoplasmic reticulum. Okay. So here, Pulal, I want to ask you one thing. What is the outermost cover of the animal cell? In the animal cell, what is the outermost cover here? Outermost cover, that means the outer. Anyone? The board of.
Cell wall, cell membrane.
Hey, my dear, look at the screen. I'm asking about regarding the animal cell, my dear. In the animal cell, the border will be plasma membrane or cell membrane. Am I right or wrong? Plasma membrane or cell membrane. What can you say about the plant cell? What is the border? What is the outermost cover of the animal cell? Plant cell?
Cell wall.
Cell wall. Just look at the picture here, the cell wall. I'm going to compare that one by using that 3D model. Right here. The outermost cover of the animal cell will be plasma membrane or the cell membrane. But for the plant cell, the outermost cover will be cell wall. Am I right or wrong? Because this will be needed for this question. That's why I explained this one. This factor, this point will be needed to answer the question in this paper. Okay. Now we'll discuss the answers now, Bilal. Okay. Here, A. What is A here? Okay. Wait a minute. Look at the picture here, Bilal. We have endoplasmic reticulum, and there are two types of endoplasmic reticulum. Here, look at the picture here. One of the endoplasmic reticulum have ribosome on their surface. Am I right or no? In their surface, they have the ribosome. So that endoplasmic reticulum is called as rough endoplasmic reticulum, RER. And here you can see another endoplasmic reticulum. They don't have the ribosome. Am I right? Or here, can you see that one? Here we have that small dot-like structure, that ribosome. But here you can see there is no ribosome. So these kind of endoplasmic reticulum are called as. Anyone, can you tell the name for that one? The endoplasmic reticulum without the ribosome. Can anyone tell me the name?
Smooth endoplasmic reticulum.
Very good, very
Smooth
good.
endoplasmic.
Very good, very good. Smooth endoplasmic reticulum. Did you get that idea, Bulal? If they have the ribosome, they are called as rough endoplasmic reticulum. If they don't have the ribosome, they are called as smooth endoplasmic reticulum. This is the 3D structure, right? Okay, now you want to study regarding the 2D model now, because in the exam, they will give the picture like this now. Okay. So I'm going to explain that RER and SCR by using the 2D model. Okay. Look at the picture here, Bulal. Here we have the endoplasmic reticulum. Here we have the endoplasmic reticulum, and here also we have the endoplasmic reticulum. Can anyone tell what is the difference that between that smooth endoplasmic reticulum and the rough endoplasmic reticulum? Can anyone tell me the difference between those two structures, Bulal?
Rough endoplasmic reticulum appears as sheets, but smooth endoplasmic reticulum appears as tubes.
Okay, that is also a very good difference. Okay, look at this picture. Okay, they can be as a sheet, a paper-like structure, and here they are tube-like structure. Smooth endoplasmic will be tube-like structure. Rough endoplasmic will be flat structures. Okay, that is also right. But Pulahal, the main difference, that is also a difference. Okay, that is not wrong. Okay, fine. But Pulahal, you can say here they have the ribosome at the surface, right? In the rough endoplasmic reticulum, they will have the ribosome. But in the smooth endoplasmic reticulum, we don't have the ribosome. If you have pimples in your face, it will be smooth or rough. It will be rough. Same like that, if they have the ribosome, it is like pimples, right? So surface will be rough. If they don't have the ribosome, the surface will be smooth. Okay. So with that idea, here, wait a minute. Okay. So here you can see there is no any ribosome. The surface is smooth. So smooth endoplasmic reticulum. But here you can see the ribosome. Can you see that on the dot-like structures? Can you see that on the rough endoplasmic reticulum will have the ribosome at the surface. That is the main difference that you want to know. So according to that, according to that, what is the name of A pull? Can anyone say what it is, a smooth endoplasmic reticulum or rough endoplasmic reticulum?
Rough endoplasmic reticulum.
Why it is rough endoplasmic reticulum, Swastika? Why it is rough endoplasmic reticulum?
Because it's plant cell.
No.
Because it has that endocarp.
Well, they have the ribosome. They have the
Because they
ribosome.
have
Can you see that dot-like structures there? Did you get that idea, Fuller? They have dot-like structures on the surface. So that's why I am telling that rough endoplasmic reticulum. Did you get that idea? How did I find that? Is that RER? Rough endoplasmic reticulum. Did you get the idea? How did I find that is RER? Okay. If you didn't get the idea, please look at this picture here. Well, this is a flat sheet-like structure. They have the ribosome at the surface. RER, rough endoplasmic reticulum. So endoplasmic reticulum become rough due to the ribosomes. But here, look at this the endoplasmic reticulum. They don't have the ribosomes, right? So they are called as smooth tube-like structure. They are flat-like structure. So in that picture, how can you find that A is rough endoplasmic reticulum? Because we have the ribosomes. Okay. And B, what is this pillar? Anyone?
mitochondria.
Mitochondria. This is, you can look at the picture here. Mitochondria. Well, you will have a small confusion between the mitochondria and between the chloroplast, because in the 3D model, okay, you can differentiate the chloroplast and the mitochondria, but in the 2D model it will be a challenge, because here, listen here, this is chloroplast and this is mitochondria. Normally in the papers they will give the clear picture, but some of the model paper, that means not government papers, if some model papers, when you're looking at that picture, definitely you will be confused between the chloroplast and the mitochondria. In the animal cell, definitely they don't have the chloroplast, no. So if there is a structure like this, definitely it will be mitochondria. So there will be no confusion in the animal cell, because animal cell doesn't have the chloroplast, no. But in the plant cell, you may have a confusion, because look at the picture here. Okay, in the picture they have mentioned, you can differentiate the structure. But some of the papers, it will be seems to be similar. The chloroplast and the mitochondria will be seems to be similar. So at that time you want to look after only one thing, Kunal. The mitochondria will be rod shape. You know what is rod shape, right? Rod shape. Wait a minute, I will explain that one. That is in my next slide. Okay. I'm going to explain regarding the mitochondria by using this picture, right? Okay. Because I haven't mentioned those things, because last class we have explained. That's why. Okay. It's okay, fine. There will be no any issues. Okay. Look at this picture, Bilal. The mitochondria will be rod-shaped structure, sausages-like structure. It will be elongated, okay? But chloroplast is not that much elongated. And Bilal, you can see here, the mitochondria will be like this, right? Not that much bended, little bit bended. So by using that picture only, you want to identify the mitochondria and the chloroplast. You want to be very careful in that one. Okay. Now you will find the answer here. This B. You can't say chloroplast, because this is a animal cell. This is not a plant cell, no. So definitely you can't say chloroplast. So in this picture, there is no any confusion because clearly you know this is an animal cell, not a plant cell. So here definitely that will be chloroplast. No, sir, that definitely that will be mitochondria, not the chloroplast. So B will be mitochondria, not the chloroplast. And E, what is this plural? Some of you have mentioned.
Nucleolus.
Some of you have sent me the answer nucleus, and some of you have sent me the answer nucleolus. Bilal, you want to be very careful, Bilal, because clearly they have mentioned the nucleolus. You may remember, right? Inside the nucleus, we will have the nucleolus, right? A dark structure. So that is not nucleus. You want to say nucleolus. Okay, Dani? Fine. Question number 2. What is a structure does not present in the cell, in this cell, but present in the plant cell? You know, already explained by using the difference. Okay, they have given the picture of the animal cell. Definitely here you can say this is animal cell. Why Polar? This is animal cell. Can anyone tell why this is animal cell?
They do not
Because of the
contain
cell wall
It does not have the cell wall. Very good. It does not have the cell wall.
and also the vacuole.
It doesn't have the vacuole. Very good. Yes, we have explained 4 differences, right? So in the plant cell, we will have the cell wall. But in the animal cell, we don't have the cell wall. And in the animal cell, we don't have the chloroplast. In the animal's plant cell, we will have the chloroplast, right? Second difference. And third one, we have big vacuole. But in the animal cell, we don't have the big vacuole. So in this picture, you can identify clearly, you can say this is an animal cell because they don't have the cell wall, they don't have the chloroplast, and they have the, what is the name given for this one, do you remember? I said that can be found only in the animal cell, but not in the plant cell. You know,
centriole.
centriole. Very good, centriole. Okay. So these are the 3 main things that I have been used here to identify that this is an animal cell. Okay. So what is a structure does not present in this cell but present in plant cell? You know, that is chloroplast and cell wall. Okay. Now we are going to discuss the functions. Okay. First one, regarding the function of the mitochondria. Can anyone tell me, Pulal, what is the function of the mitochondria? I told 2 functions and you want to mention 2 functions together. Can anyone? What is the function of the mitochondria?
Aerobic respiratory reaction take place within the mitochondria to release energy.
Reading a sentence that is given in the okay. Okay, listen, hey, Pulal, listen. Pulal, don't make it hard, my dear. Okay, your answer is correct. Don't.
For the metabolic activities of the cell.
Right. Okay, Pulal, listen. Hey, don't make it hard. Make it simple. Okay. By aerobic respiration, they will produce the energy. For producing the energy by aerobic respiration, because the main
Metabolic activities.
the energy is produced for the metabolic activities. Energy is producing for the metabolic activity. The function will be like this, Pulal, producing the energy. So because here some of the students will be confused like this, Pulal. There is a reason to combine those 2 functions, that producing the energy by aerobic respiration. I said earlier, no? So there's a reason for that one. Because in the exam, if they are asking what is the function of the mitochondria, you can write producing the energy. But in the exam, one of the model paper, they have given like this: aerobic respiration. What is the organelle responsible for the aerobic respiration? So at that time you will be confused when you have memorized only producing energy, producing energy, producing energy. So you will be confused at that time who is the one responsible for the aerobic respiration. Am I right or wrong? So that's why I said that mitochondria is the one who responsible to producing the energy by the process of aerobic respiration. You may have a doubt. What is aerobic respiration? That will be discussed in unit number 8. So when they are asking, when you're memorizing, when you're studying this one, you want to memorize like this. Function of the mitochondria will be producing the energy by aerobic respiration. And also, Pula, don't make this lesson hard, okay? We have only 10 organelles and 10 functions. And that cell division part. So don't think that this lesson is hard. Make it easy, okay? Don't make it hard. So here, producing the energy, production of the energy. Who is our responsible for that one, Pulahal? Mitochondria. If the question is like this, aerobic respiration, what will be the organelle, Pulahal? If they ask aerobic respiration, mitochondria, same. By the aerobic respiration, they will produce as the energy. Okay. Now the second one, Pulahal, regarding the cytoplasm, because in that question they have given the function, but they are asking the organelle. Okay, I have told the answer. Okay, sorry. Okay. Okay, listen here, Pulahal. In this structure, you can see here, inside the jelly, we have the nuts, fruits and the nuts. Am I right or no? Inside the jelly, we have the fruit and the nuts. Same like that, Pulahal. In the cell, there is a gel-like structure. Inside that gel-like structure, all the organelle has been submerged. Look at this picture here, Pulahal. Look at this picture. Here, all the organelles are not floating or not flying in the atmosphere. They have been inserted or they have been submerged into a jelly-like medium. That medium is called as cytoplasm. Did you get that idea? What is cytoplasm? Cytoplasm is a jelly-like liquid. Here you can see in the gap they have filled cytoplasm, a fluid within the cell. So that in that fluid, in that jelly-like medium, all the organelle has been submerged. Okay, Tane. So with that idea, look at this picture, Pulahal. This is jelly. Inside the jelly, we have the fruit and the nut. Same like that. Here we have the jelly-like medium. What is the name given for the jelly-like medium, Pulahal? What is the name of that gel? Not gel, that liquid?
Cytoplasm.
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 So you want to reply me, okay? Then I will be very, very happy, right? When you are replying my questions, okay? Because this is a physical, not a physical class now. So you want to reply, then only I can confirm, okay, my full padikiranga அப்படின்னு சொல்லி. Okay. So this is a liquid, that cytoplasm. So in that liquid, all the organelle has been submerged. Okay. Now we are going to discuss what is the function of that organelle. Functions of the organelle. So function of the cytoplasm, that is bearing the cell organelles, carrying the cell organelles. Wait a minute. So they are the ones who are responsible to carry the organelle. Am I right or wrong? These fruit and nut are carried by the gel, right? Same like that, not gel, jelly. Same like that, here the cytoplasm is the one who is responsible to carry all these organelles. So bearing the cell organelles. And second one, maintaining the shape. Pulahal, you know that one in the animal cell, we don't have the cell wall, right? In the plant cell, we have the cell wall now. So that's why they have a unique shape. Am I right or wrong? They have a unique shape. It will be like this or like this. They have a defined shape. But look at the animal cell. It seems to be like irregular shape, right? When you it seems to me like that they don't have a defined shape. Am I right or wrong? Because they don't have the cell wall. But this cytoplasm will help to maintain the shape. Am I right or wrong? This is jelly now. A jelly-like liquid, it will be supporting them to give a shape. Did you get that idea, Pulahal? Yeah, you can see here that there's a jelly, so they have a shape. Am I right or wrong? They have a shape. Same like that. This jelly-like medium, that cytoplasm will help to maintain the shape. And third one, carry out metabolic process. You may have a doubt, this metabolic process, what are the examples? Those part will be discussed in future. But for the grade 10, as a grade 10 students, there is no need to worry about that one. Sir, will the animal cell? No. Animal cell will never change the shape. But the organelle which is responsible to give the shape, that is cytoplasm. Okay, my dear. They will never change their shape. Okay, Muhammad Farhan, did you get the idea, my dear? Right. Now, come to that question here. Okay. So, bearing the cell organelles and carry out different metabolic process. Which organelle is responsible for this one, Bilal? What is the organelle responsible for this one?
Cytoplasm.
Cytoplasm. Okay. Now, third one. Can anyone tell me who is the one responsible to transport the proteins, Bilal? Who is the one responsible? Pardon, louder.
Rough endoplasmatic reticulum.
Very good, mashallah. Okay, fine. Rough endoplasmic reticulum. And now I want to ask, I will explain, don't worry, I will explain, I will explain, okay? Those who don't know the answer, wait a minute, I will explain. Can anyone tell me, who is the one responsible to produce the proteins? Transporting
Ribosomes.
ribosomes. Very good, Hashmita, very good, okay. Now we will discuss regarding the R here, don't worry, yeah? Here RER, rough endoplasmic. Already we have discussed it on that when they are marking the organelles, you may remember that one, right? So here we have the ribosome at the surface. So that ribosome is there that attached to the RER. You know, Bulahal, those who have missed the last class, okay, I'm very sorry, okay, I can't explain that one again. Keep that one in your mind that ribosome is the one who is responsible to produce the protein, okay? Here we have the ribosome. Those ribosomes are the one who are responsible to produce the protein. So they will produce the protein, and this RER, rough endoplasmic reticulum, will transport the protein. Did you get that idea? You can easily memorize that one. Ribosome is attached to the RER. Ribosome will produce the protein, and that produced proteins are transported by the rough endoplasmic reticulum. Did you get that idea, Buller? Here we have the ribosome here, and also this ribosome will produce the protein, and this RER will transport the protein. Okay. Now you know the function for that question, right? Okay. So transporting the protein is done by RER. Can anyone tell me, Buller, what is the organelle responsible for this one? That producing the secretory substances?
Golgi complex.
Golgi complex. Very good. Okay. Can you tell me there is another two function of the Golgi complex? Producing the secretory substances, okay. And other than those, that function, there are two other functions.
So
Packaging them and transporting.
Very good. Packaging and secretion. Very good. Well, those who don't know the idea, don't worry, okay. Those who don't know the idea, don't worry. I will explain once again. Don't worry, yeah. Okay. Look at this picture. What is happening here? Look at this picture. What is happening here? We have the Golgi complex here, right? We have the Golgi complex here. So these Golgi complex has been producing these vesicles, okay? That secretory substances are produced. You can observe here that they have been packed some kind of things inside that package, that vesicles, that secretory substances. So they have produced that secretory substances, and they have packed that unwanted things, and they are we can't say transporting, okay? So they will secrete that one out of the cell. Can you see that one here? So here production of the secretory substances, okay. Here production of the secretory substances, okay, and packaging, pack those things, and secretion. Did you get that idea, Pula? Functions of this Golgi complex: producing the secretory substances and packaging and secretion. Did you get that idea? The function of the Golgi complex.
Yes, sir.
So in this question, we have learned four things, Pula. What is the function of the mitochondria? Producing the energy by aerobic respiration. What is the function of the Golgi complex? Packaging and secretion and producing the secretory substance. What is the function of the vacuole, Bulahal? Anyone? Vacuole, that a big organelle was there in the plant cell. Vacuole, not vacuole, sorry. Cytoplasm. Can anyone tell me what is the
water balance, support the explosion of color to the cell by pigment within.
Right. Very good, very good. That is function of the vacuole. Sorry, I haven't explained that one. No, I'm very sorry. I'm asking regarding the cytoplasm. Jaza help me. The answer was correct. Okay, very good. The answer was correct, but I'm asking regarding the cytoplasm. Can anyone tell me the function of the cytoplasm?
Maintain a shape of the cell.
Maintaining the shape of the cell, bearing the cell organism, carrying. Very good.
Carry out different metabolic process.
Yes. Very good. Now I will continue with the next question, right? Okay. Here, based on the practical from unit number 6, the first practical that observing under the microscope, that practical. Okay. So can you answer this question, my dear? Okay. Sir. Okay. Arfa Ashik, very good, my dear. Amna, okay. And Jaza Helmi, your answer was correct, but I have made a mistake. Okay, I'm very sorry. Okay. And Sheshani. Okay, okay. We are getting. Yes, my dear. I will be sending the recordings. The YouTube link will be shared in the group. Okay. Do this question, my dear. Don't take more time. Okay. Within a short period of time, you can do the question here. That is not that much hard. Sorry, sir. We were absent to this session. Okay. Okay. Raiha and Rahul. I will. I am recording those sessions. Okay. Once again. That not live class recording. I am recording again in a different way, right? So I will be sending those recordings. Okay, Raiha. Don't worry. Yeah. So this work plan is really helpful as a reader. Okay. Welcome, my dear. And this is a paper class for grade 10. Tomorrow you will have the paper class. Grade 11 students will have the paper class on Thursday. This is a paper class for grade 10. Okay. So you can join for that class also. Venushi. I don't know whether I'm pronouncing the proper way. Okay. Shashani. Okay. Hikmar is one. Send me the answers. ACDB. Right. Even you can send me the answer through the WhatsApp, right? I will be in the WhatsApp, so you can send me the answers through the WhatsApp. No any issues, right? If you are feeling difficulty to type, you can send me the answer through the WhatsApp. No issues, right? And don't take more time, Pulal. Raha Mariam. Tana Raha Mariam, right? Tomorrow you will have the exam, right? Right. Of course. Pulal, if you don't know the answer, you can refer the textbook again. No any issues, okay? And Okay. So Bilal, look at the screen, my dear. Some of you has sent me the answers. Sorry. And some of you are being silent. I don't know why. Okay. Fine. Bilal, look at the screen, my dear. Look at the screen. Now I'm going to explain. Right, okay. Now we'll discuss the answers, Bilal. Don't do any other works. Now your time is over. Now I'm going to explain the setup, right? Okay. And Bilal, the same question, this question was asked in last year support seminar. 2025 support seminar was released by the government. So in that paper, they were asking the same question. This is a model paper from Northwestern Province from 2020 or 23. I forgot the year. But the same question was asked in that final paper, that support seminar paper in 2025, last year. So these practicals that are from your unit number 6, they are very, very important part. You can't ignore those things when you're becoming grade 11. Normally, when you're becoming grade 11 students, they will ignore these parts, these biology parts. No, you can't skip those things, Pula. They are very, very important part to your final exam. So keep that on in your mind. Okay. Now we'll discuss that practical, right? Okay. So here, to observe the any plant cell, we are going to take a small part from the leaf. Not the leaf, from the onion peel, but in the picture we want to take the sample from the onion peel, onion skin, but here I have put the image related to the leaf. Okay, but actually we want to use the onion peel, and we want to take that sample and we want to put into the water. This is not a beaker, okay, petri dish, small cup like structure will be in the lab, petri dish. Okay, this is not a bucket, small petri dish, cup like structure. Okay, so we want to put some water, we want to put that sample into the water like this. We want to dip that one into the water. So did we had the same question in our first? Okay, yeah, normally they will ask this practical in our first term and second term papers. Okay, and we want to take that sample and we want to place it into the glass, right? This is called as glass slide, right? This is called as glass slide. And after we want to put some water, we want to add some water and we want to cover it by the cover slip. Here, you can see here, this is cover slip. This is called as cover slip. This is called as the glass slide. So we have taken the glass slide and we have kept the sample between the glass slide and the cover slip and we have covered that. Did you get the procedure, Bilal? The same procedure will be followed for the animal cell also. We want to take the sample from the cheek and we want to add some water and we want to keep that sample in that water and we want to cover the cover slip. The same procedure will be followed for the plant cell, but in the plant cell we want to keep that sample in the petri dish here. And we want to take that sample and we want to keep that in the glass slide and we want to cover it by the cover slip. Did you get that idea, Pulal, how to prepare the sample for this practical? Can you raise your hand those who have any doubts in this practical setup?
Excuse me, sir.
Oh yes, Tasmita, Solomon. Tasmita.
Sir, petri dish and watch glass are same or different?
Petri dish and watch glass both are different things, Pulal. Okay. Wait, I will show you the image for the petri dish and the glass slide. Petri dish will be cup. Wait a minute, I will show you the picture. I can't explain that one. I will show you the picture for that one. This is petri dish, right? You can use both of them, no any issues. But in that watch class, the curved structure will be easy to put the sample. Wait, I will show you that now. Did you get the idea, my dear? This is petri dish. This one, and this is the glass slide. That glass slide.
So
Watch
the
glass.
base in the petri dish is flat. Is it so, sir?
Yes. Watch glass will be little bit bent, curved. Okay. Did you get that anyway?
Yes, sir. Thanks.
And wait for a while. I'm going to show you the question that they asked in the 2025 support seminar paper. Wait a minute. You can see the screen. Here, this is the question that they were asking in that support seminar paper. The same practical has given in this question, but in the question that I had shown you the model paper, in that question they didn't give the picture, but in the support seminar paper they gave the picture. That is the difference, but all the other questions were similar. The same question was there. This question have the picture, but in the question that I showed you now, it does not contain the pictures. Okay, that is the only difference. Okay. Now we'll write the answers for that question, right? Okay. Sorry. Okay. Now we'll answer the questions again. So they are asking what is the order, right? What is the order that I want to follow here? First of all, we want to take the onion peel and we want to put into the watch glass that water, and then after we want to put that into the glass slide and we want to cover it by the cover slip. Am I right or wrong? You may remember that one, right? Onion peel and we want to put it into the watch glass and we want to keep that sample and we want to cover by the cover slip. Okay, Dane? Now that question is over. And second question. What is the expected aim of D? What is the purpose of keeping that onion peel into the water? Because Polahar, the onion peel will be dry. Am I right or wrong? Onion sample will be dry. Am I right or wrong? So to make it wet, to make the sample wet, we are going to put that sample into the water. So when they are asking what is the aim of putting that onion peel into that water, you want to say to make the sample wet, or to prevent it from drying. Already it is dried, so to make it wet, that is the most proper answer. To make the sample wet, to make the sample wet. Okay, question number 3. What is the advantage of covering the tissue by the cover slip? Okay, can anyone tell Pula? What is the purpose of covering the sample by the cover slip? Why do we want to cover the sample by the cover slip? You want to keep the sample here. Pardon, Hafsa, louder.
To prevent from moving.
To keep it protected.
Keep it protected. Hafsa, I couldn't say. Hafsa.
To prevent moving, like the cells and all moving.
The move 1 hour, now mood
To prevent the entering of the microbes like.
Okay. Pull up. Listen. Shall I explain, Pull? Hamza, look at the scheme. The idea is correct, but it is a little bit wrong. Okay, idea was correct. Okay, fine. Okay, but that is not happening like that. Okay, don't worry. Pull, listen here, Pull. The purpose of covering the sample is, why are you covering the food with the lid, Pull? You were cooking in the kitchen, right? Then why are you covering the food by lid? To prevent from other organisms, am I right or wrong? So the dust particles may be enter into the sample. So when you're observing under the light microscope, you cannot observe the sample when the dust is there. Am I right or wrong? So to protect the sample from the dust and other things. First one. And second one, Pulal, when you are keeping in an open area, what will happen? The sample will dry. Water will be evaporated and sample will dry. So to prevent from drying, we are going to cover the sample by the cover slip. That is the purpose of covering the sample by the cover slip. Okay, Apulahal, did you get that idea? So what is the purpose of covering the sample by the cover slip? Means you can say to prevent from drying. To prevent from drying, or we can say to protect the sample from the dust. If the dust is there, Apulahal, the picture will be not clear. Did you get that idea? Hafsha, is it clear, my dear? Hafsha, is it clear, my dear? Hafsha?
Yes, sir.
Okay. What about others, my dear? Some of you has told the answer. Normally in the past, in the final paper, even in the model paper, even in the first term and the second term, normally students are losing the marks in the structure part. In the structure, you want to write the accurate answer. If the answer is wrong, there will be no any the flexibility allowed middle. No any flexibilities. You want to write the correct answer, accurate answer. Question will be very, very easy. You want to write the accurate answer, then only you can score more marks, right? So be very careful when you are writing the answer for the structure part. And Pulahal, the fourth question. For already we have discussed this thing many times. Today itself I have explained 3 or 4 times. Difference between the animal cell and the plant cell. Only end is a plant cell, plant is an animal cell is an animal cell. So there is no need to explain that one deeper and deeper and deeper. And question number 5, draw the rough sketch. Okay. This is a sketch that you can observe for the animal cell. This is the sketch for the animal cell. And this is a sketch for the plant cell. So you want to draw the structure like this. Some of you have drawn the structure in the proper way. But Pulahal, you haven't labeled the picture. So you will lose your marks. This is the way that how you are losing the marks. So you want to draw the diagram and you want to label the structure. In the past paper, there is a question that is from a grade 11 part. They have told them to draw the muscle tissue. So in that year, they gave the mark in the scheme. You can see that part. They have given the marks for 2 things. One for the shape, that spindle shape, that is for a grade 11. But look after the thing that how they have given the marks for that one, right? So they have given 1 mark for the shape and another marks for the nucleus and third marks for labeling them like this. First mark for the shape, second marks for the nucleus, third marks for the label part. So when you are drawing the diagram, please label them. You know that is nucleus, you know that is cell wall, but those who are correcting the paper, they don't know those things. When I'm correcting the paper, I don't know which is nucleus and which is cell wall. But you know which is cell wall and cell membrane, because you are the artist. You are the one who know which is cell wall and cell membrane. So you want to mark those things. Okay, Pulal. So in the exam, be careful, Pulal. When they are asking you to draw the diagram, you want to draw and you want to label the diagram. Okay, Tane. So with that idea, we'll mark it. This is the animal cell. And this is a plant cell. And you can see the plant cell, they have a defined shape. Am I right or wrong? Because they have the cell wall. Animal cell, they don't have the defined shape. They don't have the regular shape because they don't have the cell. Am I right or wrong? Okay. So with that idea, we'll discuss this part very quickly, okay? Because we have only limited amount of time. Can anyone tell me the function of the Golgi complex? We have already discussed, Pulal. Golgi complex.
Producing secretion.
Production of secretory.
Producing the secretory sub. Very good, very good. Hafsha, just started. Very good.
Packaging.
Packaging and secretion. Very good. Producing the secretory substances and packaging and secretion. Synthesis of protein. Who is the one responsible for that one, Pulal?
rough endoplasmic
Pullal. Producing
Ribosomes.
ribosome, Hafsa al-Qureshi, transporting will be done by Arya. Sariya Mahal. Rough endoplasmic reticulum will transport that one. Okay, my dear. Synthesizing will be done by ribosome. Be careful in that one. Okay, my dear. Right. Ribosome. And for the mitochondria. Anyone?
Aerobic, respiratory, and metabolic activities.
Jazahilmi. Okay. Hey, Mahal. Don't say that metabolic activities, okay? For the metabolic activities, they are producing the energy. Producing the energy for the metabolic activities, okay? Producing the energy is the function of the mitochondria. Producing the energy by which process, Bilal? Anyone? By which process? Jazahil, can you tell my dear? By the process of? Aerobic respiration. By the process of aerobic respiration, they will producing the energy. So, producing the energy by aerobic respiration. Okay, Babulal, okay. So in this class, totally we have discussed all the things, but we haven't discussed regarding the cell division, right? Regarding the meiosis and the mitosis, this part we haven't discussed. In our next paper class, we will be discussing the mitosis and the meiosis, and we will discuss all these questions and the past paper questions related to the cell division. Okay. So Abulal, now we are going to conclude the class. Do you have any doubts to be asked? Anyone, Bilal, do you have any doubts to ask?
Excuse me, sir.
Oh, yes. Hashmita, salome.
Sir, peroxisomes are there, no, sir?
Pardon, my dear, pardon. I couldn't hear you.
Peroxisomes.
Peroxisome.
Peroxisomes, sir.
Peroxisome, yes. That is not included in your O-level syllabus. It will be discussed in your A-level syllabus. Okay.
Okay.
They have
sir.
explained only regarding the ribosome, lyoxysomes, peroxisomes. It will be discussed in the A-levels. Lyoxysome and peroxisome. Not in your O-level syllabus. So there is no need to worry but in some of the model paper, they were asking those questions. Don't worry about that. Oh, yes, my dear. We have the tunes. Remind me tomorrow, inshallah. I will give you the tune for the grade 10. Okay, Khadija? Inshallah. Okay. So, Pula will conclude the class with the recitation. Subhanakallahumma bihamdika ashhadu an la ilaha illa anta astaghfiruka wa atubu ilaik. Assalamu alaikum wa rahmatullahi wa barakatuh. And good night. Have a nice day. And everyone. We'll meet in our next class, right?
Thank you so much.
Thank you.